another question.
Either only one gene, like MARFAN, or we do what we call. Well, this is the standard opening slide for my university, out at Amsterdam University Medical Centers. This is the adapted version for during pandemic. We still have to wear face masks in the laboratory, and this is where I come from. In Amsterdam, on the bottom left, that's my house with a group of my collaborators and students, and I officially retired there in 2016, but I still have what they call a 0-hour appointment there. I'm still active in research there. And on the right, you see a team in Prodia that I work with since 2000. I have been working with since 2018, and we develop genetic testing, but that's a bit difficult now because most of the laboratory has been shifted to COVID testing. Okay. So, Mendelian inheritance. Who was Mendel? Gregor Mendel lived in the 19th century. So he was born almost 200 years ago, and he was a German monk, and he studied in the gardens of his cloister inherited properties of peas. And on the bottom left, you see his photo. That's around the time he had his first publication in 1866 in the Naturforschende Verhandlungen. That's the German, which means the proceedings of the Nature Research Association. Actually, photography had just been invented at that time. The first cameras. This is probably made with one of the first cameras. So it's a long time ago. And what Mendel did was he looked at properties of peas. I don't have the slides for that, but it's and he found some regular properties and inheritance of properties, and he derived laws from that. So there's the law of dominance, and in that time they started using the words alleles and genes already, long before DNA was known to contain those properties. And that's really interesting. So if the two alleles at one locus differ than one, the dominant allele determines the organism's appearance. The other, the recessive allele, has no noticeable effect on the organism's appearance. And then there's the law of segregation. The two alleles for a heritable character separate or segregate during gamete formation and end up in different gametes. Now, nowadays, we know that's not always true, because if two genes are close together, they can be in linkage disequilibrium and they can inherit together.
I don't know actually what happened. Sorry.
Okay. So we have the law of independent assortment. Each pair of alleles. Okay, yes, it's recording again. Okay. So each pair of alleles segregates independently of other pairs of alleles during gamete formation. Now that's sometimes not true if they are close together, of course. So in 1953, Watson and Crick discovered the double helix structure of DNA, which you see in the photograph on the left. And I always mention an interesting thing that this was a single-page publication. It's one of the most impactful publications of the last 70 years, especially in the field of genetics. And it was published on one single page. So if you are writing publications, think about that. So after that, we began to understand how and why Mendel's laws work most of the time. Genes that carry out properties consist of DNA. Variations in the DNA cause differences in properties, and some variations or new mutations, they are pathogenic and cause disease. So inherited diseases are caused by mutations in genes. So we need to know about DNA, RNA, and protein. We need to know how to detect mutations or variations. And we need to know about the interpretation of those. And for that, we need to know the effect on the protein, and because of that, also the effect on disease. So, a reminder, DNA is double-stranded, RNA is single-stranded, and we go, just took this picture from Game of Thrones, I think. This is double-stranded, this is single-stranded, and the central dogma in molecular biology is DNA encodes RNA encodes protein. Well, normally, I hate dogmas because they make you stupid, but this is mainly true, but not completely, as I will show in the next slide. So the protein coding genes is only 1.5% of the human genome, which contains about 25,000 genes. Transcribing this to the RNA yields 100,000 or more than 100,000 transcripts, because as we discovered much later, one gene can be spliced into the messenger RNA in different ways, and that causes multiple transcripts. And from that we go to the proteome, which is more than 1 million proteins, because changes in the proteins make different proteins. Antibodies are a lot of those million. We can make more than 1 million antibodies, actually. And that's because the the gene is rearranged in ways that causes new proteins that can be used as antibodies against, for example, virus. So mutations may lead to aberrant protein or less protein. And then, some 12 years ago, we discovered that there are also genes that, we already knew that there were genes that encode only RNA. We have the microRNAs and we have the long non-coding RNAs. Now, some of those we have known for a long time, because ribosome, that's the factory in the cell that creates the proteins, that synthesizes the proteins, consists of 2 very large RNA molecules. But those RNA molecules, those RNA genes, contained 3 times more information than the 1.5% of the genome that encodes protein genes. So there is still a lot of the microRNAs are involved in regulation of the protein genes. The long RNAs, many of those we do not know the function. I have some ideas of that about that I want to investigate, but I think the instincts and other properties that cannot be encoded by protein are located in the RNA molecules. But that's for another lecture. Next slide. Genes in a genome. So the gene is the information for one trait. That's the definition from the time of Mendel. And now we call a gene the DNA code for a protein or an RNA molecule. The genome is the entire code of all chromosomes. The human genome consists of 3.2 billion base pairs. I already mentioned this, 1.5%, 4.5%, and 25,000 protein coding genes. I don't know the exact number of RNA coding genes. Variants in the DNA. So 99.9% of the DNA sequence is identical between humans. So on the average, we have 3 million variants compared to standard. So even these very different people. So between human and I like the facial expression of these two, but between a human and the closest relative in the monkey world is 99% identical sequence. And here, I'm not sure there is a difference, but I bet that to you. So the 0.1% of the DNA sequence that's different between humans consists of copy number variations, that's large deletions and insertions, We were just talking about thalassemia, where the large deletions are causing disease sometimes. But there are also large deletions and insertions that do not cause any change in properties. We have the single nucleotide variants. We call those SNVs, or the old term is SNPs, SNPs. But polymorphism implies high frequency, and there are no effect on the phenotype. But on the DNA level, there is no difference between a SNP that causes a disease or a SNP that causes that's common and causes just no effect or has a small effect on the phenotype. So that's why we call them variants now. But in this presentation, I will stick to the SNPs because it's easier to pronounce than SNPs, and I think it's very common to use the term SNPs. Even in the database of variation, DB-SNP, every variant has an RS number, reference SNP, and we still call it SNPs. So the RS number, the RS has no real meaning, it's just reference SNP. So genes and alleles. Well, this is common knowledge for all of you probably. All chromosomes exist as pairs. We have the autosomes, 1 to 22. We have the sex chromosomes, X is female, XY is male. In the bird world, it's the opposite. X are female in the birds. And because the birds are the last living descendants of the dinosaurs, it's probably true that also the dinosaurs had XX females. Two copies of a gene on homologous chromosomes are called alleles. That's also known, of course. It's also, the word allele is also used for the two copies of a SNP on the two chromosomes. So for each SNP, we have a paternal and a maternal variant or allele. So diseases are caused by environment and lifestyle, for example, passive or active smoking. They are caused by genetics, and we have monogenic diseases that are caused by Mendelian inheritance. So there can be multiple genes involved in a disease, of course, but you only need to have mutation in one of those genes to have the disease. For example, breast cancer susceptibility can be caused by mutations in BRCA1 or BRCA2 or BRCA1 and BRCA2, as some people call it. But you only need a mutation in one of those genes, and then we still call it monogenic. Polygenic is different because in that case there are many genes involved in a disease or disease susceptibility. And so you have more variants to cause the disease, but each have a small contribution. And we have genetic susceptibility, that's genetics plus environment. Also, monogenic diseases are influenced by environment, for example, breast cancer. And polygenic diseases like diabetes or heart disease are also influenced by environment. That's why, and I will talk about that on Saturday on the Indonesian Society of Human Genetics meeting, about the polygenic risk score. And that's about the effect of many different SNPs on disease susceptibility, which we can use to give people advice about lifestyle changes that can help prevent the disease for which they have higher susceptibility. Here is an example of monogenic on the left and polygenic on the right. On the left, we have an example of the recessive case where there are two carriers that have one child here with recessive inheritance, but there's one gene that causes 100% or almost 100% of the risk for this disease. And for multiple genes, we have a complex inheritance pattern. We can see sometimes, like here on the right, that the disease is present in many generations. And usually people think about, well, here you see it looks like it's skipping a generation. Well, and there are many folk tales about that. But usually it's because there is not a single gene involved in the disease, but it's more complicated. The effect of the mutations on the protein quantity or quality may cause the disease. So, and we have several types of mutations or variations that can cause the disease. We have missense, that's the change of amino acid. The effect depends on the type and the position of the mutation. We have nonsense, that's the gain of a stop codon in the wrong place. We can get a truncated protein, that is a severe effect. Or we can have loss of the mRNA by nonsense-mediated decay, and that sometimes gives a milder effect, but not always. And we have synonymous mutations that are in the coding sequence or silent. So there's no effect on the protein, on the code protein sequence, but these mutations may have an effect on splicing and therefore also on the protein. We can have in-frame deletions or insertions that causes a loss of amino acids or a gain of amino acids or the gain of a stop codon. And we can have loss or gain of a number of bases that is not a multiple of 3. And that causes a frame shift. And in those cases, most of the time, the mRNA is destroyed by the ribosome because it will detect a stop codon. And after that, it will detect another spliceosome, which is still bound to the first cycle of the messenger RNA processing. And then the ribosome triggers cutting of the messenger RNA. Sometimes frame shifts cause a short protein, and that is usually toxic for the cell. So that's those are usually very bad mutations. We can have loss of one or more exons by the large deletions that can cause a frame shift or an in-frame shorter protein with a very severe effect usually. Stop gain and frame shift mutations, I already mentioned, lead to NMD, nonsense-mediated decay, mediated by the ribosome in the first cycle of translation. This NMD mechanism is a sort of fail-safe mechanism that the cell has to prevent the toxic effect of short proteins. So detection of variation, Sanger sequencing has been around for 40 years now, I think. I give an example here of the automated Sanger sequencing. Each peak is one nucleotide, and here we see an overlapping of a C and a T. The C's are blue, the T's are red. You can see the sequence here at the top, and that's heterozygous DNA variation. In clinical genetics, it was usually called a mutation if it causes disease. But nowadays we changed that because mutations are actually new events. So we use the term mutation nowadays for new events, de novo mutations that cause disease. And we use the word variant for all other variations because there is on the DNA level not a real difference between variations that cause disease or do not cause disease. And in the case we have evidence that they cause disease, we call them pathogenic variants. Here I'm showing you always should check the forward and reverse sequence because that's on the next slide. Here you can see that in one direction sequencing you see C and T, and here you see the other strand of the DNA with A and G here. So the next slide is, this may happen if you sequence in only one direction to save money, for example. Here you see a lot of N's here, like here. So we have here, 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 and here. This looks like we have 4 mutations here, 4 heterozygous variants. If you look at the other sequence, at the other strand in the reverse direction, we see only a single one of those here. And that's corresponding with the A and T here. So 3 of the mutations or heterozygous variants in this sequence are just artifacts caused by the fact that in DNA sequencing usually, because DNA synthesis is used for the sequencing, a G followed by an A, that polymerase has difficulty processing that. So you get a low G and a high A. And you see that here and here again. So that's why you get some background of things that look like very heterozygous variants, but are not. No, this shows you the real variant. And these are the other ones. So in the reverse reaction, you see only one mutation. So this is the detection of genetic diseases, changes in my lifetime. 1953, Watson and Crick discovered the double helix structure of the DNA. In 1955, the first chromosome defects were described. By that time, the quality of the microscopes had evolved sufficiently to do that. And then in 1977, we got cell sequencing, like here, or the old-fashioned way with radioactive labeling. In 1986, we got automated cell sequencing. I was actually involved in that in my lab in Michigan. We tested the first Applied Biosystems gel-based sequences there. And in 1993, we got a capillary sequencing, and in 2005, we got a microarray. And then at present, we use the next generation sequencing, which does a lot of sequencing all at once. I won't go into that too deeply because we don't have enough time for that. This is part of our next generation sequencing lab in my department in Amsterdam. It produces several terabytes of data per day, this unit. And this is my colleague, Lulu from Prodia, Jakarta, who visited me for training for a couple of months in this laboratory. We often have students coming for a master's internship or for a PhD in my laboratory in Amsterdam. So if you're interested in that, you can always ask about it. Now, example of inherited diseases. Remember, we know more than 7,000 of those diseases. Thalassemia is a very common one in the tropics because the mutations that cause that in carriers, they give some protection against, what was it, malaria? Or I think it was malaria, yeah. But anyway, we have Marfan. I will talk about Marfan syndrome, polycystic kidney disease, cystic fibrosis, and breast cancer susceptibility. Osteogenesis imperfecta will be a separate lecture because it's a very broad subject, and we have done a lot of research on that. Thalassemias are caused by mutations in the hemoglobin genes, and the inheritance is complicated due to 2 alpha and 1 beta genes. And there is a high prevalence in the subtropic countries because carriers have some protections against malaria infection. Okay. I should have waited for the next slide to say that. On chromosome 11, we have also several pseudogenes, which are very similar to the alpha and beta and delta genes, but they're not expressed properly. They don't have RNA. So these pseudogenes are very similar to the other genes, to the real genes, and they cause problems if you want to do sequence analysis of the real genes. You have to be very careful in designing the primers and also in designing of the MLPA probes that we use. There will be a separate lecture about thalassemia, so I'm not going into that right now. Marfan syndrome. It is usually autosomal dominant inherited, caused by mutations, usually in the fibrillin 1 gene. Cardiovascular, they have mitral valve prolapse, aortic root dilatation, and also aortic aneurysms and dissections. I've made those red because those are the most common causes of death in Marfan syndrome. However, the patients find the lens luxation more affecting them because it can cause blindness. They don't worry about suddenly dropping dead with an aortic dissection or rupture, but they do worry about becoming blind. Skeletal features are tall stature, long limbs, as you see here, arachnodactyly, spider hands, like we see also here, and pectus excavatum or carinatum, as you can see here. It's the carinatum. These features are not always present. So we have families in which some people look like this and some people look completely like a normal, not Marfan syndrome patient, but they still have the aneurysms. So that's why mutation detection in this syndrome is very important and combined with, also need to be combined with genetic counseling. This is Chanditse. I'm showing her picture here because she is for an Asian girl, very tall, 173 centimeters. And she has very long limbs. Her legs are 110 centimeters. Usually the length of your legs is half of your total body height. So that should be like 85, 87 centimeters for her. And she has the spidery finger. So I don't know. I have tested her, but the Marfan symptoms are highly variable. So maybe this girl does have Marfan syndrome. And when I walk in the streets in Jakarta, I often see people that look like her. Not as pretty, of course, but not always as pretty. But it's possible that we have many more Marfan patients than we think. So that's why I was looking at mutations. These are the variants that we found in our lab in the Amsterdam University Medical Centers. And this is our database. And we have this week we had 893 unique variants in more than 2,000 patients, meaning that most families have their own unique variant. So you cannot say we have found the mutation for Marfan syndrome because it's usually different every time. So it's also difficult to find out whether it's likely that this mutation is pathogenic. These are the different effects of the mutations. Frame shifts are often the cause. And then, I looked at the genome aggregation database, and I found that in East Asia, there are a lot of people that have a pathogenic or likely pathogenic variant. And I found that in this database, in 1 in 2,800 individuals. So in Indonesia, which has 2.8 million people, I expect 100,000 Marfan patients that are now not being diagnosed and not being treated. So some Marfan cases have autosomal dominant, most Marfan cases have autosomal dominant inheritance, but we have found also recessive inheritance in some families. So far we found 5 big families that have that, and that's caused by very mild mutations. So the carriers have no real phenotype. We did find some mutations already in Indonesian patients. As you can see here, this is part of my database in Amsterdam. And I visited Sulawesi once, and we have the Tanah Toraja rock raves there. The dolls in those niches here are representation of diseased people. Deceased, not with a S, with a C, of course. And then here we can see that there is in one family, they're close together, we have very tall people here, and here this one needs extra space for the legs. They made it here. So I was thinking maybe this is a family in the Tanah Toraja people that have Marfan syndrome. We never tested them, but it's possible. It's just that it could be a nice subject for research there. Because if you show this picture, and you, at the same time you find a mutation in the fibrin 1 gene in this village, it's much easier to get it published, because every scientific journal would love to publish a picture like this. Prevalence is highly underestimated in East Asia, and mutation testing of FBN1 in Indonesia could be important for genetic counseling, pre-symptomatic diagnosis, and medication. I won't go into the medication right now. The next disease I want to talk about is dominant polycystic kidney disease, or ADPKD, caused by mutations in one of these 2 genes. Age of onset depends on the mutation. It's early to late adulthood. You get multiple cysts in both kidneys, leading to kidney failure and death, usually within 5 years after the diagnosis. So DNA testing is important for genetic counseling. The recurrence risk is 50% because it's dominant. And treatment with medication is possible if you diagnose this early enough. So that's why genetic counseling is so important here. For the PKD1 gene, I found in the genome aggregation database many pathogenic or likely pathogenic variants, and 1 in 500 people is carrier in East Asia, so probably also in Indonesia. So these people will probably develop the polycystic kidney disease around age 50 or 55 very often, which will lead to severe kidney failure and death within 5 or 10 years. This is a great burden for hospitals, for families in the first place, of course, for hospitals, for health insurance, for people who lose their ability to do their job. So it's a very important issue to get this testing done. It's not simple, because it's also a very difficult gene with a huge, very similar to that gene also. So sequencing this gene is very difficult. Cystic fibrosis, autosomal recessive, it's the tough mucus disease. It's due to mutations in the CFTR ion transport protein. Undiagnosed children die young due to lung and or pancreatic problems. And according to American studies, there is no cystic fibrosis in East Asians. However, this was based on the test with a mutation panel for white European people. Most Americans are white European descent. So, in Europe, we have high frequency incidences, 1 in 3,600. Carriers are 1 in 30, probably because carriers are protected from cholera. So why don't we see it in Asia? Well, Professor Song from Kuala Lumpur, I met him at a conference, and he was trained in Australia. I told me he recognized CF in a child suspected of a parasite infection. He ordered a sweat test kit from abroad. It was not available in Malaysia. And the patient tested positive for CF. And since then, they found many more CF patients in Malaysia. And this is why in the database, we find a lot of pathogenic mutations in this gene. The total minor allele frequency is more than 0.5%. And this is an underestimation, because for many mutations that I see in the database, many variants, I do not know whether or not they are pathogenic. So we expect a lot of cystic fibrosis in East Asian, Southeast Asian countries, but they are probably not recognized because they die young of suspected parasite infection or suspected pneumonia. So my conclusions, exactly 45 minutes. Most inherited diseases are not diagnosed and not properly treated. We need awareness of this. Research is needed to assess frequencies and develop therapies. DNA testing is important for genetic counseling, recurrence risks 25 or 50%, early diagnosis and treatment, and training and accreditation is needed for clinical geneticists, genetic counselors, and scientists for research and interpretation of test results. Oh, sorry, that was my final slide. Thank you for your attention. Questions? Let me stop sharing.
Okay.
Share. Yeah.
Okay.
Share.
Okay. Thank you, Prof. Gerard. So now it's time for us to discuss and take some questions. Please raise your hand or no? Yeah. Please, if you have any question, just raise your hand. If not, then maybe I will start with the question.
There's already there's a message in chat. Maybe that's a question. I don't know.
Really?
From?
No, not yet.
No.
Okay, so maybe one question for Gerard. So, if there is pseudogenes in the gene of your interest, how usually the trick to design the primer? Because most of the time we forgot about this pseudogene.
Yeah,
so
that's a big problem.
How is the trick to design the primer?
Well, I usually use the software which is freely available at the NCBI. It's called Primer Blast, and you can design primers with that. And it will look at the entire genome for possible sites that are also being amplified. So it should take care also of pseudogenes. But if you want to be really sure, you have to look at the sequence to make an alignment of both sequences and see where the differences are. And for the PKD, we've done that.
Okay.
And that was difficult. It was hardly possible. So what we do now for the polycystic kidney disease gene is for the PKD1, we have designed primers for long-range PCR. So you can be using also differences between the real gene and the pseudogene. So you make a very large PCR fragment of several kilobases, and you do a pre-amplification with those primers. You need long primers for that to assure high specificity. And that way you can do what we call a nested PCR. So you can amplify the entire gene in two very large fragments, and then you do the PCR with the primers for each exon on that product. So you do not amplify the pseudogene, you amplify only the real gene that way. And you don't need a very effective PCR for the first side, for the long range PCR, because even if you don't see a large product on an agarose gel, it's still possible to do the nested PCR. So if you want help with that, you can just contact me.
Okay, so just normal PCR, yeah. I mean, So you do the long range PCR and then you do shorter PCR using the template, right? This is what you mean?
Yes.
Okay. Okay.
And for next generation sequencing, it's more difficult because most software will not map the fragments. The fragments are usually not longer than 100 bases, and for the PKD that's difficult. So I designed a way to solve that by removing the pseudogene from the reference genome before starting the software analysis.
Okay.
And that way, everything will be mapped to the real gene. And if we find differences that are potentially pathogenic, then we have to do the nested PCR for that exon only to see whether or not in which of the two copies of the gene it's located.
So this is the standard procedure, Prof. Gerard, that if you analyze the NGS result, you delete all the pseudogene from the reference. Is it like normal procedure, or is only you doing this?
It's only me that's doing it. It's new.
Okay. So no, I'm wondering about my NGS result then.
Well, we can if you still have the results, we can still do this. If you have kept the raw data, like the BAM and the BI file, the BAM and the BAI files, the big files, we can still do reanalyze that using the genome in which the pseudogene has been removed.
Okay.
You have to know some.
I will discuss about this later with you in more detail. So, is there any question from student Edwin or Isa? Is there any question about Mendelian disease? Not yet. Teh Lulu, Dr. Dina. Not yet. Okay, so maybe another question, Prof. Gerard, about the DNA diagnosis in Netherlands for Mendelian disorder. So I know that in Netherlands all is covered by insurance, right?
Yes.
And the default is always trios, right? So if the affected child came, it's always the DNA of father and mother you also analyze, right?
No. It's not. No, only if you do exome sequencing. For many diseases, we do either only one gene, like Marfan, or we do what we call panel testing. And for that, we have a panel of genes that are, and we do an enrichment of those genes before we do the next generation.
So you do not run the trios, only the patient. Only the patient.
Yeah, for some diseases, there are so many genes that we want to run the trios. For example, for primary ciliary dyskinesia, that's the disease of the lungs, of the cilia. We already know more than 40 different genes. And in so many genes, in so many patients, there is no mutation in those genes. So then we run trios with exome sequencing or gene sequencing.
For thalassemia, for example, how normally you do it in Netherlands? Or is not exist anymore, thalassemia in Netherlands?
It is still present, yes. We have many, because we have many immigrants in the Netherlands from tropical areas and from the Mediterranean area.
Yeah.
So about 10% of our current population is immigrants.
Yeah.
So that's a lot.
So you still have thalassemia in the Netherlands?
Yes, we do. Yes, and we just use Sanger sequencing combined with MLPA. That's simpler than doing the next generation sequencing.
So also trios? No, only the patient.
Only the patients. Yeah. And then after that, when we know the mutation, we can just do PCR of the exon involved, or just do the MLPA test.
Yeah.
MLPA test is not an expensive test. It's only like for the entire gene, it's I think it's about 10 euros per sample or something like that.
Yeah, in Netherlands, but in Indonesia you have to multiply by 3, Prof. Gerard, or 5.
Well, we managed finally to get the government to give tax exemption for research purposes. If you want, I can send you the forms. It's
tax. For the tax.
you can import materials tax free now.
Okay.
I've been lobbying for that for 10 years, together with some people from colleagues, from former colleagues from the universities of Gajah Mada and of Samara and Undip, like Professor Ali Gufron. He was for a long time. He has been, and now not anymore, but he has been the Director General of the Department of Higher Education and Research.
Yeah.
And he helped me with this, and also some other former professors of Undip and Gajah Mada and the University of Jakarta are in the government now. So, and if I know them, I can just send them private emails.
Yeah, I mean, they should know our struggle, right? But yeah.
No, but
the condition is still the same.
Yes, but it's
just wondering.
no, but we can get tax exemption, so we can get tax-free import of. I will send you the forms for that. We can. It saves a lot of money.
Yeah. Okay. So maybe is there any comment from Prof. Ani or Prof. Ramdan, Prof? Prof. Ani? So Prof. Ani and Prof. Ramdan, they're also their research focus is also in thalassemia. So a lot of people, let's say people in Faculty of Medicine of Pajajaran. Our focus, let's say, in genetic research is more to thalassemia, because like 40% of thalassemic people live in West Java. So if the total population of thalassemia patient is like 10,000 in Indonesia, 40% of them live in West Java. So the disease burden is quite high. So yeah.
But most of the people of Indonesia live in West Java, isn't that true?
20% of people of Indonesia live in West Java.
No, but the problem in Indonesia is also that there are so many subpopulations. We have still some more than 300 different recognized ethnic groups in Indonesia that may have their own private mutations that cause the disease.
Yeah.
Because the selection due to cholera epidemics has been the same in each population. But in Jakarta and the other big cities, these populations are mixing, of course. Still, in the rural areas, they don't mix. They marry their own ethnic group.
Yeah. So another question. Still, there's no question from student. Okay, so another question for Gerard. So if you analyze the mutation in, let's say, exon intron boundaries, how far will you go in the intronic region? And do you always do the functional study for the mutation you identified in the non-coding region? Because could mean nothing, like in the non-coding region. So how you usually do?
Oh well, I have a separate lecture about that. It's an hour, but no, it's still.
This is our struggle in thalassemia research. Let's say we design our own primer for exon 1, 2, and 3, and also the promoter region.
But there are several steps in that.
But the thing is, there is still mutation in the middle of the intron one. Let's say like minus 604, I think. In the intron one.
Yeah.
So how can they find this mutation? Is it they just sequence the whole?
No, before we had next generation sequencing and genomic sequencing, we just sequenced the RNA.
Yeah.
Because these intronic mutations can be found only by looking at the RNA.
Yeah.
And so then you can, if you see what the effect on the RNA is, then you can see, you can start looking for the mutation in the intron. That's how this mutation was found long ago.
Oh.
But, and if you look at intronic variants now, because with genomic sequence we see a lot of those, of course. We first go to the genome aggregation database to look at the frequency in the population. So if the frequency of a dominant disease is above of the mutation in a gene that causes a dominant disease, It depends on the frequency of the disease, of course. But then I usually take the limit of 1 in 1,000. If it's above that, it's already likely not pathogenic. And for recessive, you have to take the limit a lot higher because the most common mutation causing recessive cystic fibrosis in Europe has a frequency of 3% in the population. That's yeah.
So
and
1 in 1,000, do you mean that the MAF that you use for cutoff in dominant trait is 0.1%?
Yes.
Oh.
In the American Society, the American College of Medical Genetics, they take 5%. And
yeah,
I think
that's what we use.
And that's far too high. If one single mutation can cause a lot more than the entire population frequency of that disease, the level of 1% cutoff is fine.
Okay.
I'm writing a publication about that at the moment. So I already mentioned it in my study, in my review about Marfan syndrome that was published 2 years ago. So in one of the Indonesian journals, the Journal of Medical Biology or something like that. I don't remember exactly the name of the journal. Mm-hmm. It's published in Semarang.
Okay.
I can send
you. So then you conclude directly that this is more pathogenic variant, or you said that assuming this is a pathogenic variant?
No, it's the other way around. If the frequency is above 0.1%, I assume it's not pathogenic, it's benign.
Yeah.
And if it's rare, that's not evidence of pathogenic effect. It could still be a neutral variant that's not pathogenic and is rare. So for pathogenic effects, we have software called Alamut.
Yeah, Alamut.
By Sophia Genetics, and you do a lot of studies, and we do a lot of work in that. There's also several, and there's one important issue here. If you look at algorithms for prediction of pathogenic effect, there is one called SIF, S-I-F, which uses Grantham distance. Those are ignoring all changes of charge. So for most mutations that cause a change in charge, they are completely useless. So if you use SIFT, S-I-F-T, you will always get conflict of conclusion. And then the American College of Medical Genetics says there's a conflict between the conclusions of different software algorithms, so you cannot use that. So it's not that you can say it's a VOS then, a VOUS, and that's stupid. And I'm going.
So usually I use like Polypen2 mutation tester and SIF. So I use 3 because Alamut has three software together, right? This Alamut.
Shift. I don't use SIFT anymore. It's too bad. It's too
okay.
bad. Yeah, I use mutation tester.
Yeah.
And Polypen. And then I use my brains because I have a degree in protein.
Yeah, student, my brain is as yours, so I still depend on the in silico program.
Yeah. No.
So maybe Prof. Ramdan, maybe is there any comment from you, Prof? Or maybe he's not present.
Yeah, I can see his.
Yeah.
Ramdan Panigora.
Yeah. Yeah. Sorry, Unia.
Yeah, Prof. So sorry. Good afternoon.
How are you, Prof. Bas? How are you?
Hi, thank you.
Fine, thank you for your presentation. I think it's very. I appreciate it and enrich our appreciation about genetics, but the details will be handled by Unia and the gangs. But thank you very much, Prof. Bas. Thank you.
Yeah, thank you, Prof. Ramdan. Prof. Ani, is there any question regarding thalassemia research, Prof. Ani? Because Prof. Ani is one of the key person working on thalassemia, and also Dr. Susi. So a lot of people from thalassemia group in this in this meeting. Dr. Susi or Prof. Ani, is there any comment? But yeah, for sure, we have to discuss separately about thalassemia project. We sometimes discuss with Prof. Sutat, but maybe also to be good to discuss with you about the technique in the lab that we struggle. We are still struggling until now. Yeah, especially for hemoglobin A, we didn't do much about the mutation analysis for hemoglobin A. I mean, thalassemia alpha, let's say. Yeah.
Okay, yeah.
So, student, maybe Senia, Edwin, is there any question from you? Anything, you can ask anything. Don't think about that. Wow, my question is, sometimes you don't think like, oh, my question is so stupid, but there is no stupid question, We will never give you a stupid answer, right, Prof?
Oh.
Not give any stupid answer. So Edwin or Iza, Salbia, silahkan kalau ada pertanyaan.
Patricia.
Shafira, kalau ada pertanyaan.
I have a question.
There's a question.
Yeah, silakan.
Yeah, Professor, thank you for
the
Professor, thank you for the very interesting presentations. In your previous slide, you mentioned that the Human Genome Project has identified about 6% genes that encoded the protein and RNA. I just wondering, what about the rest or the other 94%? Is that consists of untranslated region or what? I mean, the huge number of those nucleotides must have certain function, right? Okay, thank you, Professor.
Yeah, there are many variants where it's very difficult to predict the effect, for example, in the untranslated regions. And that's why in such cases, we need to look at the RNA. We need functional evidence of the effect of the mutation. That's really a big issue. Most laboratories, when they do panel testing or even exome sequencing, they ignore the untranslated exons even. So many genes have 1, 2, or 3 extra exons which encode the 3' untranslated region. And usually if there are more than one, there's alternative splicing of those regions that's involved in the tissue-specific expression of the gene. And that's a region. And if there are splice errors in the splice sites of those exons, we don't see them because the most laboratories don't look at that. And that's an issue that's very important. You're right about that. And the sequence variants in those exons, it's very hard to predict what they do. We look at the binding sites for specific enhancers or proteins that are involved in gene expression. And but if the binding sites are changed, we still need functional evidence. So we do a lot of work with cell culture to do that, because you need cells that express the gene to see the effect on the RNA or on the protein. So that's it's important. But we haven't completely solved yet.
Okay, Professor, thank you.
You're welcome.
Is there any other question? Last question, maybe from student. Iza or Shafira? No. Maybe last one from me, Gerard. So regarding the non-coding RNA. So in your slide, you showed that for 4.5% actually we have gene encoded non-coding RNA, right?
Yes.
And I'm just wondering, like for example, in Hirschsprung disease, even though with a lot of attempt to find the gene, we only can identify gene in, let's say, in total cases only 40%. And the rest we call it like missing inheritability. So now people start thinking maybe this is like, defect in expression of mRNA. So we do also, study in RNA, but it's not really proof a lot of things to answer this missing inheritability. So I'm just wondering, from your experience in osteogenesis imperfecta, whole exome sequencing, for example, is how many percent that you identify mutation in the gene encoded non-coding RNA for Mendelian disorder?
I don't know, because we haven't done whole genome sequencing yet for osteogenesis imperfecta. We're still looking for proteins, looking
Okay.
whole exome sequencing. And
So,
then if you find something, it's very difficult to prove, because we don't know what the variations in the RNA coding, in the LNRNA coding genes, non-coding genes are doing, what the effect will be, etc. So it's really difficult.
So you need whole genome sequencing, of course, yeah, to
If
know
you want to look at the
things.
RNA genes, yes. And
because, for example, for Hisprung, there's only one article showing that there is actually mutation in the gene encoded microRNA. So, maybe the missing inheritability is actually there,
Yes, many microRNAs could be involved in disease, of course. And
but not in the level of expression, Prof. Is in the gene itself.
Yeah, that's why not now in some diseases we do now RNA-seq, so sequencing the total RNA, and then you look at all the genes and also at the expression differences. So you see, in RNA-seq, you can look at the mutations, but you can also look at the expression.
Okay. That could be a nice approach, right? To see whether there is mutation in the.
But for RNA-seq, you need cells that are involved in the disease. So we developed now methods to derive cells from the fibroblasts, skin biopsy from the patient. And we can do direct transformation of those cells into smooth muscle cells for study of Marfan, or we can do osteoblasts or chondrocytes for other diseases. And then we can look at the RNA. And we already use that for to look at the functional effect of mutations, because the gene is not expressed in fibroblasts or in blood. For example, MYH11 is involved in aortic aneurysms.
But are you how sure are you that the pattern of the expression in the IPS is exactly the same with the DC cell, let's say?
We don't use iPSCs.
Oh, you don't use? Oh.
Human fibroblasts still have the potential to transform into many other cell types. So we don't have, because iPSCs are very expensive and take a long time. From human fibroblasts, we take 3 weeks to make osteoblasts.
Oh, okay. So you directly differentiate from fibroblast to osteoblast. Okay.
Yeah, the direct transdifferentiation. And for osteoclasts, it's not possible. We either have to go to iPSCs or we have to take blood. But you need 50 mils of blood to have enough precursor cells to create osteoclasts. But there's a lot of work.
50 mils, quite a lot.
This sounds like something we need to have more often discussions about research here with you. We can do that online if you want, with the professors and the medical.
That's interesting. I mean, for osteogenesis, I can imagine that you do that approach. For Hisprung, it's quite difficult. You have to get the biopsy from the colon, and then you culture the ENS for genital.
It's going more difficult. Yes.
Okay. Okay. So, if there is there any question? Oh, our student is so silent today. Okay, so anyway.
Still awake?
So next week we will have another lecture from you, right, Prof. Gerard?
Yes.
So more about genetic of osteogenesis imperfecta. So maybe next week also our student from biomedical sciences will join, and also maybe people from the Department of Orthopedy, because their research is about osteogenesis imperfecta will join. Maybe five people that focus on osteogenesis imperfecta, that will give us enlight, let's say, the approach on how you do research on Mendelian disease.
I will include some part about how we discovered the new gene and how we proved.
That's what we would like to learn, Prof. So how you do the gene hunting in the Mendelian disorder, how this the strategy, and
this can be. You can use it as an example for any other disease.
Yeah, that would be nice because thalassemia is also nice, but I mean it's more complicated, right? It's monogenic, but it's really complicated.
Yeah, it's really complicated.
It's really complicated. Okay, so if there is no other question, thank you very much, Prof. Gerard. And if you don't mind, can we get your slide and I will upload it in our student website, Live Unpad, we call it.
Yeah. Just give them also my email address.
Okay. You already sent me to my email, yeah? Then I will. Let's say.
I have sent your email, yes. And if you send it to the students with my email address, then they can contact me if they have questions.
Okay.
Yes.
Okay. Yeah. Thank you very much, Prof. Gerard.